to the Chapter 9b podcast on chromosomal inheritance. If you haven't watched the 9a podcast on genetics or Mendelian inheritance, you should probably go back and look at that. In that, I describe Mendel's ideas, the laws of segregation and independent assortment, and he really got it right. The only thing he didn't really understand was chromosomes, and so that's what I'm going to kind of talk about in here, and then a few other modifications to his theory. So, in this picture right here, this is Thomas Hunt Morgan. He worked at Columbia University, and he sorted out how genes and chromosomes are related, and he did that using this, which is Drosophila melanogaster, or the fruit fly. I love the scale here. So this is like 1 millimeter, and so these things are really, really tiny. But this is just a regular. We sometimes refer to that as wild type Drosophila. So this is just run-of-the-mill fruit fly. The other picture I included in here is this guy, which is Sturdivant. He's one of the people who worked in Thomas Hunt Morgan's lab, and as an undergraduate, he figured out a ton of stuff, and I think that's a really good kind of a lesson. I'll talk about him in just a second. Okay, so in Mendel's experiments, the big one, if you remember, was that cross he made between purple and white to create purple flowers, and then those purple flowers, once they would self-fertilize, he got 3 purple, but then that white came back, and so it really came to that idea of what is a gene and what is this thing that is kind of skipping a generation and comes back. And so here's the experiment, and kids tend to get lost on this in my class, and it's really not that confusing, so try to stick with me. What he did in this cross is he has a heterozygous, gray body, long-winged fruit fly. He crossed that with a black body vestigial wing. And so this has 2 mutations. One is it's black, and one it has these tiny little wings. And these things are hilarious to watch them try to fly. They just can't, so they just fall. Anyway, in a cross like this, where it's a dihybrid cross between a heterozygous for both traits and a homozygous recessive, what Mendel predicted, in other words, in a Punnett square, what he predicted is that you should have an equal number of each of these different types of fruit flies. In other words, you should have 1 or a 575 of these gray long, 575 of these black vestigial, and then 575 of each of these if we were to have like 2,300 fruit flies. In other words, we should get a 1 to 1 to 1 to 1 ratio. These ones are actually called recombinants because they don't look exactly like the parents, but these two do. So what did Thomas Hunt Morgan figure out when he did this actual cross? He found something weird, and so there's another discrepant event, and so he has to kind of go back to the drawing board. What he finds is that there are way more of the parental types than there are of these offspring. And so it's the first time we have to look back and say, wow, did Mendel really get it wrong, or did he not thoroughly understand it? And so what they finally figure out in studying this for a while is that these 2 genes, in other words, the gene for gray body and long wings and the genes for black body and vestigial wings, didn't sort independently. In other words, they were tied together, or they were linked. And so a way to think about that is the gray body and the long wings, if we look on the chromosome here, those 2 alleles in this female are on the same chromosome, and so they're linked together, and so they traveled together. They didn't sort independently. Same thing with the black body and the vestigial wings. They stayed together. And so what we discover is this idea of recombination, or frequency of recombination. In theory, this number should have been 50%, and if we ever get a number then that is less than 50%, that means that they're on the same chromosome. I don't know if you can see my hands, but the closer that number gets to 0, the closer they are. In other words, if it was 2% recombination, that would mean that those genes are right together on the chromosome. And if it was 0, that would mean that they're the same gene. And so the cool thing they were able to figure out back then is that by using all these frequencies of combination, they were able to figure out where the genes are on the chromosome. And it was actually Morgan's student as an undergrad who took all this data home and figured out this whole idea of gene maps, which to me I think is a pretty cool story. Okay, here's some other patterns of, in other words, we could say modifications or tweaks on the ideas of Mendel. First one is the idea of incomplete dominance, and so the one you'll always read about is this cross between red and white snapdragons. They produce pink. Now you should immediately think, wow, that sounds a lot like blending, but it's not blending. In other words, this one is not totally dominant over the white. And if you do a cross of the pinks, what we eventually get back are red, pink, and white. And so these 2 genes are neither dominant, or it's incomplete. And the cool thing about that is you get a phenotypic ratio of 1 to 2 to 1, as well as a genotypic ratio. Next is the idea of codominance, and a great example of this would be blood groups. As far as blood groups go, we've got carbohydrates on the surface of the blood, and those come in, we'll say, two flavors. We've got the A carbohydrates and the B carbohydrates. And so in blood, you can either have no carbohydrates on your surface, and you'd be type O. You could have just the A, just the B, or you could be type AB. And so this is another modification. You don't have to only have just one or two alleles. You can actually have multiple alleles, and those can be codominant to one another. Next one is the idea of pleiotropy. Pleiotropy is when you have one mistake or one mutation, and that mutation creates a whole bunch of characteristics. So this would be, it's actually one single letter that causes this hemoglobin mutation, and that creates these blood cells that actually look like that, which can lead to all these different side effects of having that kind of a mutation. Now the flip side of that is this. This is polygenic inheritance. Polygenic inheritance is when instead of you just having one gene, we've got multiple genes. And what you'll find is a lot of the examples that your teachers give you about what is one gene, or they'll talk about like a hitchhiker's thumb or a earlobe that falls down, and those are really obscure examples, like a widow's peak. And the reason why is that there really aren't that many traits that we have that are caused just by one gene. Most of them are caused by polygenic inheritance. So skin color, for example, you have a number of different genes, and those genes play out all of these different phenotypes. And so we have this characteristic bell-shaped curve of different traits that you could have. So your height, I remember reading once, is caused by 13 different genes spread across all these different chromosomes. And so it's really hard to predict what that would actually be. Okay, the last pattern I want to talk about is this. If you're like me, I'm actually red-green colorblind. I can't see anything in this picture. I can make out some kind of brownish, but I really don't see what the shape is. I do know that this should be a picture of a sailboat. And so you could laugh at me and say, oh, poor colorblind Mr. Anderson. But really what's going on, let me show you what's going on, is this. This is me right here. In other words, I'm a male who has color blindness. And so if you're ever dealing with traits that are sex-linked, you have to include the sex chromosomes. And so let's trace back. My father is actually unaffected. That means on his X chromosome, he has one good copy of that gene that allows us to make the correct cones in our eyes, and so we can delineate those colors. Now my mother has one bad gene, we'll say, and one good gene. Is she colorblind? No, and that's the cool thing about being a girl. You can have one bad gene over here, but you've got a backup copy. In other words, we can think of this as a recessive trait on the X chromosome. So let's play out the 4 different types we could get. This right here would be a boy who is getting his X chromosome from his mom, but it's a good one. This would be a girl who gets 2 good X chromosomes. This would be a girl who gets a good and a bad X gene. And then this would be me. And so what's funny is I actually have a sister who is a carrier of that gene. And that means that she's not colorblind, but her son or my nephew is colorblind. Not one. I think there are 3 of them, and only one of them is colorblind. And so it's cool to trace that. But when you're ever trying to figure out sex-linked traits, remember that you have to include the X and the Y